Good morning, everyone. Today I will discuss about the part 2 of overview of mycology. In the last session, we came to know about the taxonomy and classification of fungi. This is the fungal classification and taxonomy. Now, the latest one, we learned there are 10 phyla. Now coming to the phyla one by one. The first recap of the medically important fungi: Ascomycota, Basidiomycota, previously Zygomycota, and Chytridiomycota. So this is a chart. Ascomycota, they have septate hyphae. Spore type is ascospore. Example Candida, Pneumocystis, Histoplasma, Aspergillus, etc. Main feature is they have cup-like sacs, or those are called asci. Basidiomycota, these also have septate hyphae. Spore type is Basidiospore, and examples are Cryptococcus, Trichosporon, Malassezia. They have club cells. Zygomycota with coenocytic hyphae. They have sporangiospores, example Mucor, Rhizopus, and Absidia. They are fast growers. And now we have seen that Zygomycota are not monophyletic. They are divided in different phyla, and the name uh, has been changed. Chytridiomycota, they also have coenocytic hyphae. The spore type is zoospore, example Allomyces, Neocallimastix, and they are flagellated. They are motile, unicellular with chitin cell walls. Now, the few important facts about Chytridiomycota. These are the earliest diverging, uh, these are from earliest diverging final fungal lineage. Chytridomycota are basal fungi. They diverge first from the fungal lineage. Fossil and molecular clock evidence places their origin around 600 million years ago. These are only true fungi with flagellated cells. They produce motile zoospores with a posterior single whiplash flagellum adapted to aquatic or moist terrestrial habitats. Now, mostly they are microscopic and unicellular or filamentous. Some are unicellular, others have simple coenocytic hyphae. Thallus type, monocentric, polycentric, and holocarpic forms exist. They are parasitic and saprobic lifestyle. Saprobic means they break down tough materials like chitin and keratin, and parasitic means they infect algae, diatoms, protists, and amphibians. The Batrachochytrium is the example of pathogenic species. They cause chytridomycosis in amphibians. The disease is a major cause of global amphibian decline. Now, Batrachochytrium dendrobatidis, in short form capital B, small d. This is the first fungus known to drive vertebrate species to extinction. Reproduction, both sexual and asexual. Asexual reproduction via zoosporangia producing zoospores, and sexual reproduction through planogametes or resting sporangia. In some species, nuclear cycle is haplontic, means zygote is the only diploid stage. Now, habitat and ecology found in ponds, wetlands, soil, dung, and decaying plant material, often isolated using bait techniques like using cellulose or chitin. Phylogenetics and classification placed in supergroup Opisthoconta related to Blastocladiomycota and Neocallimastigomycota. Current classification includes 9 orders: Chytridales, Rhizophidiales, and Spigellomycetales. The molecular features: Genomes of BD and B cell are compact but encode unique expansions of protease and cell wall modifying enzymes. Now, what is BD? As I have told, Batrachochytrium dendrobatidis is BD, and B salamandrivorans is the B cell. So BD and B cell, they have compact genetic codes with unique expansions of protease and cell wall modifying enzymes. Lack full mitochondria in some lineages shared with Cryptomycota and Microsporidia. Research used model organisms for evolutionary biology, cell motility, and fungal flagella research studied for bioremediation, biocontrol, and host pathogen interactions. Example, allomyces, they are the model organism for alternation of generations. Batrachochytrium dendrobatidis, pathogen of amphibians. Rhizophidium, they are algal parasite, and Chytridium species, they are saprobe or parasite. Now, next, latest important facts about Neocalimastygomycota. They are strictly anaerobic fungi found only in the guts of herbivores and herbivorous animals, especially ruminants like cows, sheep, elephants, etc. Cannot survive in oxygen, so they are obligate anaerobes. They lack mitochondria. Mitochondria are absent. Instead, they contain hydrogenosomes, organelles that produce hydrogen gas and ATP anaerobically. They have multiflagellated juice pores. Unlike most other fungi, their juice pores have multiple posterior flagella. These features sets them apart from Chytridiomycota, which have only one flagella, not multiple flagella. They have enzymes through which they can degrade fiber highly effectively. They possess extremely potent cellulases, xylinases, enzymes capable of digesting tough plant materials like cellulose, hemicellulose, and lignin. Those are critical for ruminant digestion. They are mutualistic symbionts form symbiotic relationship with their host animals, contribute to nutrient acquisition, especially volatile fatty acids from plant matter. Their taxonomy and diversity, they are under phylum Neocalimastygomycetes. Mycota includes one class, that is the Neocalimastygomycetes, and families are Neocalimastygaceae and Sycomycetaceae, etc. Genera Neocalimastics, Pyromyces, Aneromyces, and Orpinomyces. Genome adaptations, small AT-rich genomes. Horizontal gene transfer HGT from bacteria observed, especially genes for plant polymer degradation they have. Genome studies reveal loss of mitochondrial genes and expansion of carbohydrate active enzyme families. Now, they have unique morphology. That means coenocytic thallus with rhizoids. Rhizoids means root-like structures. Sporangia release multiflagellated juice pores in the gut lumen. They are found in gut of herbivorous animals. Life cycle is asexual. No confirmed sexual stage have been found yet. Ecological and biotechnological significances. Now, studied for biofuel production due to their powerful lignocellulose degradation, potential use in bioreactors, animal nutrition enhancement, and bio waste conversion. Now, what are the recent discoveries? New genera and species isolated from Asian elephants, giraffes, yaks, bison, and hindgut fermenters. Ongoing research in synthetic biology and engineering of CAZ enzymes for industrial use. CAZ means carbohydrate active enzyme families. Now, example, genera and host, Neocalimastics found in cow rumen, Pyromyces elephant horse, Orpinomyces in sheep, goat, and aneromyces in buffalo and camel. Now, what are the differences between Chytridomycota and Neokalimysticomycota? Habitat Chytridomycota, as I have told, they are aquatic or mostly terrestrial. They are the most important cause of amphibian decline. And these Neokalimysticomycota strictly anaerobic gut environments of herbivores they love. Oxygen requirement, Chytridomycota aerobic or facultative anaerobic, but obligate anaerobes are Neokalimysticomycota. Spores, Chytridomycota produce uniflagellate spores, and these are multiflagellate Neokalimysticomycota. Flagella, single posterior flagellum, Chytridomycota, Neokalimystico, multiple posterior flagella. Mitochondria, Chytridomycota have functional mitochondria, whereas Neokalimysticomycota lack mitochondria. They have hydrogenosomes instead. So chytrids swim, but Neo chews. It's an important mnemonic. Nutrition, chitin based, the typical fungal saprophytic, and their cell wall of Chytridomycota are chitin based, that is typical fungal. But this is Neokalimysticomycota, this is also chitin based, but not those of typical fungal. Now, nutrition, saprophytic and parasitic on algae, amphibians, and this is mutualistic, digest cellulose in ruminant gut, herbivore animals. Reproduction, both sexual and asexual. This is only asexual cycle known. Sexual cycle is not yet confirmed. Now, thallus type, unicellular or simple coenocytic. This is coenocytic or rhizoidal. Genome features, moderate sized classical fungal genome. This is compact, AT-rich horizontal gene transfer from bacteria. This is important. Enzyme production, moderate plant breakdown by chytrids, and for neos, it is highly cellulolytic, powerful lignocellulose degrading enzymes are present. So, in short, Chytridomycota are aquatic fungi with single flagella, whereas Neokalimysticomycota are gut fungi with many flagella and huge digestion powers. Now coming to the Rosella species, now included in phylum Cryptomycota, also known as Roselomycota. Placement, basal or earliest diverging lineage of true fungi in clade Holomycota. Why those are important? Because it was once a mysterious genus with unclear classification. Molecular phylogenetic studies, especially rRNA gene sequencing and phylogenomics, reveal that Rosella represents the earliest diverging fungal lineage. It lacks chitin in some life stages, which is very unusual for fungi. That's why it's mysterious. Now it's the model genus for the Cryptomycota or Roselomycota phylum. In the previous day session, this slide I have shown where Rosella species came very first. So this was the earliest diverging lineage. And Neokalimysticomycota and Chytridomycota, they were included in some same lineage. Later on, they were separated. So for Rosella species, this is mysterious uh, model genus for Cryptomycota, Roselomycota phylum. Now, what are the key characteristics of Rosella? Habitat, obligate endoparasites, example infective chytrids, oomycetes. Chitin present only in spores, not in vegetative stages. Chitin is absent in vegetative state. Flagella, they are present like chytrids, posterior flagella cell wall, often absent in some life stages. And reproduction, again asexual via zoospore, sexual cycle not well documented. So for Chytridomycota, both are there. For Neokalimysticomycota, only asexual cycle, and for Rosella also only asexual cycle. Nutritional mode, intracellular parasites they are. Now, one important phyla, that is the Microsporidia phylum, that is the Microsporidia, distinct phylum of obligate intracellular parasites. Not true fungi in the classical sense, but closely related to fungi. They have features some like parasites and some features like fungi. Based on molecular phylogenetics, they are now considered part of the Holomycota clade, which include fungi and fungus-like organisms. So key features: habitat obligate intracellular parasites. In animals, especially insects and fish. Spores produce tiny, highly resistant spores with a polar tube used to inject their contents into host cells. It is a special feature. Polar tube to inject contents into host cells. Cell wall contain chitin like true fungi. Mitochondria, they lack classic mitochondria, have mitosomes instead. So Neocalimysticomycota have hydrogenosomes and Microsporidia have mitosomes. Genomes extremely compact, among the smallest of eukaryotes. Reproduction mostly asexual via spore formation. Now classification debates were there. From traditional viewpoint, their placement was in Protista, but no longer it exists. Recent placement from molecular viewpoint, related to fungi or within fungi, especially close to Roselomycota or Cryptomycota. Current consensus: early diverging fungal related lineage, Holomycota, sometimes treated as a sister phylum to fungi. So till exists, but is now seen as an early diverging lineage related to fungi, not a classical fungus like Ascomycota or Basidiomycota. So, what are the differences between Microsporidia and true fungi? This is single-celled, Microsporidia single-celled, and true fungi, they may be single-celled, maybe multicellular. It lacks true cell wall, but fungi have chitinous cell wall. Microsporidia reproduce through asexual cycle only. True fungi, they have diverse lifestyles. Those are the obligate parasites, Microsporidia. But fungi, all are not obligate parasites. Few are saprobes, few are mutualists also. And these lacks mitochondria. True fungi have mitochondria. Spores with polar tube to inject the materials into host cell. The spores without polar tube and present in true fungi. So, this much for today. Thank you. Hope you enjoyed the session. Don't forget to like, share, and subscribe. Thank you.